Hi, my name is Emily Fraser, and I have been working with Dr. Kramer for these past few semesters as well as this past summer to investigate the morphology and decline of Hawaiian land snails. So, our focus is on the Hawaiian Islands as they have a large snail diversity with more than 700 unique species. And specifically, we chose to focus our project on the genus Acatanella, or Oahu tree snails. These organisms are ideal for this project for a number of reasons. So, species on Hawaii have been relatively isolated for millions of years. The species introductions are very rare, and so it makes it easy to track the impact of invasive species on the decline of communities. And the reason this occurs is because the Hawaiian Islands, the only way to enter or exit the islands is by swimming or flying, and these land snails are unable to do either. Acatanella also act as an indicator species, and historically they do not respond well to changing conditions. And this is mainly due to their small population sizes, and they're also very particular about their preferred habitats and produce very few offspring. This makes it so they can indicate the overall health of ecosystems. And what we learn about extinction in this group may also apply to other imperiled species. Acatanella also play an important role in the Hawaiian culture. Many poems and artwork refer to these snails and their habitat. And typically, this poetry falls into the love genre, so they talk very positively about these organisms. One example of this is singing snails, also the shell that sounds long is one of the examples. And so many native chants and stories also refer to these snails and their habitat in a positive manner. Acatanella also play a large role in Hawaii's ecosystem. They are present in the nutrient cycling system, so essentially they recycle the environment, returning nitrogen to the ground, and have been able to provide food for fungi and algae growing on leaves. They also act as decomposers, so they break down dead material and allow it to be recycled back into the environment. And they are very essential to the energy flow throughout the ecosystem. So what we already know about the causes of decline in this population is that habitat destruction due to farming and climate change have played a very large role over the years. But also humans have introduced the carnivorous land snail, also known as Euglandia rosea, and they introduced this snail from Florida in an attempt to lower the population of the giant African land snail. And this is because the giant African land snail was eating farmers' crops. But this kind of backfired, and the carnivorous land snail quickly depleted the native populations of snails on the islands, and this included Acatanella. There's also been a spread of non-native vegetation by humans, and the lei industry has also played a role in the decline as they have been collecting shells prior to the mid-20th century. And what we are trying to investigate throughout this project is which species are disappearing and what does this decline indicate about the morphological diversity of Acatanella. So what we've been focusing on through our materials and methods is Dr. Kramer went to the Bishop Museum at the University of Hawaii in 2016, and he was able to photograph their collection there. And we standardized these photographs, and then over this past summer, I was able to take these images and identify the shell outlines using ImageJ and a Mica Toolbox plugin. So these images were then further standardized using color standards within each photo, and then they were scaled to size. We then further converted the images to cone catch, and what that means is we took the image information and simulated how it would be perceived by human photoreceptors at a distance of about a half a meter. After we completed this, we underwent the QCPA framework, which is known as the Quantitative Color Pattern Analysis, and we performed this on each plate. And what this was able to give us information on was the luminance, which is an estimate of the brightness of the shell, color intensity, which is an estimate of the shell color saturation relative to that grayscale, and also the within shell color diversity, which estimates the shell color complexity. And so with the help of Dr. David Sisko, we were able to estimate the year that each species was last seen in the wild. And with this data, we were able to use the R statistical analysis platform to look at patterns of decline between the morphological traits of Acatanella and the years that they were last seen in the wild. What we found from this investigation is that Catanella species have disappeared at a steady rate throughout the 20th century and into the 21st century. And there seems to be a directional shift near 1960 in remaining shell shape variation, luminosity, color, intensity, and within shell color diversity. So before the 1960 shift, we saw declines in luminance which refers to how white the shell is. So luminous and poorly pigmented species were declining before 1960. Also, unusually shaped species were seen to be declining at a higher rate, and there was also a decline in shell shape variation before 1960. Once this 1960 shift occurred, we saw darker, more heavily pigmented species disappearing as mean luminance increased again. The standard deviation of luminance did not increase as well. So this information is indicating that the mean and standard deviations of the remaining living species are changing. So we also see that average shape species after 1960s are disappearing, and the morphology of remaining species becomes increasingly dissimilar. So increased variance before 1960, as variance and standard deviation decreased, the species dying off are found on the edge of morphospace. But around this 1960 shift, standard deviation and variance begin to increase and continue to do so throughout the 21st century. This suggests that species towards the center of morphospace are disappearing rather than those at the edge of morphospace. So our big takeaway from this is that there's a possible shift in the driver of species decline around 1960. And based on our past research, it is possibly due to the introduction of the carnivorous land snail, Euglandia rosea, which was introduced in 1955 in that effort to protect farmers' crops. So this snail hunts using the slime trail, but some populations of Catanella may be more vulnerable than others. So our next steps in the future is that we are hoping to complete statistical analysis and evaluate the hypotheses that 1960 marks a meaningful shift in the pattern of decline in a Catanella. We're also hoping to expand the scope of these analyses to include the remaining species of a Catanella. And our hypotheses for the future are that unusual morphologies are preferentially lost, leading to rapid declines in morphological variation faster than expected. We're also hypothesizing that average morphologies are preferentially lost, preserving the morphological variation even through ongoing extinction. And our null hypothesis throughout this investigation will be that declines are indiscriminate, leading to the gradual loss of morphological variation with ongoing extinction. I would like to give acknowledgements to some of the people that we could not have done this project without. So we do have the help of some of our colleagues in Hawaii, as well as this project was supported by the CURAS Teens Award this summer. So we'd like to give thank you to all of them, and thank you for listening to this presentation.